Today we're diving straight into a really fascinating scientific detective story. We are going to go figure by figure, panel by panel, to decode exactly how the loss of a major Alzheimer's disease gene, SORL1, systematically breaks down the brain's immune cells. By the end of this, you're going to understand the microscopic cellular trafficking of Alzheimer's disease like an absolute expert. Let's get right into the data.
Here is a super quick roadmap for today's explainer. We'll start by defining the SORL1 microglia mystery, and then we'll logically walk through the paper's data step by step. We'll hit cargo and degradation in Figure 1, enzyme activity in Figures 2 and 3, the CIMPR trafficking defect in Figure 4, accumulating cargo in Figure 5, and we'll wrap the whole thing up with exocytosis and inflammation in Figure 6.
All right, Part 1, the SORL1 microglia mystery.
Okay, before we arrive at the actual cellular crime scene, we need to make sure we're strictly comparing apples to apples. Throughout this study, the researchers are comparing healthy, wild-type stem-cell-derived microglia with isogenic SORL1 knockout cells. And we gotta define our terms carefully right out of the gate. SORL1 is the actual DNA gene, right? While SORLA with an A is the functional sorting receptor protein that gets produced from that gene. So if researchers create a SORL1 knockout, they are disrupting the gene, which means the cell entirely loses the functional SORLA protein.
Now, what does SORLA actually do? Well, think of the cell as this massive, bustling city. The Golgi apparatus, that's your packaging and shipping center. The endosomes are the local sorting stations, and the lysosomes are the degradation and recycling centers. In this city, SORLA is basically the cargo manager. It's a transmembrane sorting receptor, and its whole job is to make absolutely sure packages or proteins travel to their correct intracellular destinations.
And we've studied SORL1 a ton in neurons, but its role in microglia is a completely different ballgame. Neurons use this machinery heavily for receptor recycling, but microglia? They are professional phagocytes. They are literally the brain's garbage trucks. They are constantly engulfing cellular debris, dead synapses, protein aggregates, you name it. Because they eat so much, all that material has to eventually be processed by lysosomes. So losing SORL1 in microglia isn't just a simple recycling issue, it creates a massive lysosomal processing defect.
Which brings us to our first piece of hard evidence. Figure 1, cargo and degradation.
So the researchers needed to know, can these knockout cells still digest what they eat? To find out, they set up what's called a DQ-BSA pulse chase experiment. It's really clever. BSA is just a model protein cargo, but DQ-BSA is heavily labeled with a fluorescent dye. When the protein is intact, the dye molecules quench each other, so they're totally dark. But when lysosomal enzymes finally chop that protein up, the dye molecules separate and glow brightly. So step 1 is the pulse. Load up the cells with this cargo. Step 2 is the wash. Wash it away to stop any new cargo from entering. And step 3 is the chase. Track that glowing fluorescence over time to see exactly how well the cells are degrading the food they already ate. And the results here are just wild. When we look at the data using a different fluorescent tag just to measure uptake, it clearly shows the SORL1 knockout microglia actually swallowed up more cargo than the healthy wild-type cells. But when we check the DQ-BSA degradation, the knockout cells produce significantly less fluorescence. So what does that mean? It means they are aggressively eating more garbage but completely failing to digest it.
This naturally leads us to Figures 2 and 3, enzyme activity. Right. Seeing this massive digestion failure, you have to ask, are the lysosome's digestive enzymes actually broken? They tested 3 different enzymes to check. Cathepsin B, which is a cysteine protease that cuts up proteins. Cathepsin D, an aspartyl protease that works in highly acidic environments. And HEXB, which doesn't cut proteins at all. It actually removes sugar groups from complex molecules. By checking all 3, they were looking for a broad, system-wide lysosomal defect, not just a single broken protein. And sure enough, fluorometric assays confirmed the catalytic activity for all 3 of these enzymes was significantly reduced in the knockout cells.
But here is where it gets really interesting. It's a total paradox. A Western blot, which measures the total amount of a protein, shows that the enzyme levels in the knockout cells are perfectly normal. Think of it like a factory. The Western blot tells us all 10 workers showed up for their shift today, but the activity assay tells us those 10 workers are basically getting zero work done. The enzyme proteins are physically present, but their actual catalytic activity is severely crippled. So, why?
Well, fluorescent tracking shows us exactly where these workers are. The enzymes aren't broken, they are just lost. In the knockout cells, the enzymes strongly overlapped with TGN38, meaning they were stuck back at the trans-Golgi network, the city's shipping center. Conversely, they showed way less overlap with LAMP1, meaning they were failing to reach their final destination down in the late endosomes and lysosomes. The workers are in the building, for sure, but they are stuck in the mailroom instead of working on the factory floor.
Let's jump to the next piece of the puzzle. Figure 4, the CIMPR trafficking defect.
This whole biological traffic jam introduces us to the missing link, CIMPR. That stands for cation-independent mannose 6-phosphate receptor. Going back to our city analogy, if the Golgi is the shipping center and the enzymes are the packages, CIMPR is the vital delivery truck. It literally physically transports those tagged enzymes out of the Golgi and down to the endosomes.
In a healthy cell, this process is an incredibly precise mechanical relay race. Step 1, enzymes get synthesized. Step 2, they get tagged with M6P address labels. Step 3, SIMPAR binds to those tagged enzymes. Step 4, SIMPAR drives them away from the Golgi toward the endosomes. Step 5, the enzymes are released into the acidic compartments to do their job. And finally, Step 6, the empty SIMPAR delivery truck is recycled back to the Golgi to pick up the next load.
But take away the SOLA cargo manager, and this entire supply chain completely collapses. In the knockout cells, the total amount of SIMPAR protein goes way down. And on top of that, significantly less of the remaining SIMPAR actually reaches the downstream delivery zones. The delivery trucks are either totally missing or they are completely failing to finish their routes. And that perfectly explains the massive trafficking defect. Without SIMPAR, those enzymes are never going to reach the lysosomes.
All right, moving on to figure 5, accumulating Alzheimer's cargo. This is the actual crime scene. We need to see the real-world consequences when these defective microglia are forced to face actual Alzheimer's cargo. The researchers fed the cells fibrillary amyloid beta 142, literally the aggregates you find in Alzheimer's plaques, and synaptosomes, which are just fragments of neuronal synapses. And because the knockout cell's appetite far exceeds its broken enzyme-starved degradation system, this highly dangerous cargo heavily accumulates inside the compartments. The garbage is literally just piling up inside the cell.
Now, I want to make a really crucial clarification here, because we have to interpret this precisely. Accumulation just means the uptick of amyloid and synaptic debris is exceeding the cell's ability to clear it. It does not mean 100% of the cargo goes undigested. The knockout cells still have a tiny sliver of baseline function left, but they are massively overwhelmed because their molecular supply lines are totally broken.
And that brings us to our final set of data. Figure 6, exocytosis and inflammation. So what happens when a microglial cell is stuffed to the brim with garbage it can't digest? Well, it tries to spit it out. To measure this lysosomal exocytosis, the researchers used a brilliant trick with the LAMP1 protein. Think of the lysosome as a balloon. The specific N-terminal domain of LAMP1 lives on the inside surface of that balloon. It only ever sees the outside environment when the lysosome physically fuses with the cell's outer membrane and pops open to release the garbage. By using an antibody that only grabs that specific inner domain from the outside of the cell, researchers can literally count how many lysosomes are successfully fusing and spitting things out.
And the aftermath for the knockouts is pretty grim. The data shows reduced surface LAMP1, meaning the cells are failing at exocytosis. They also showed a much lower extracellular release of those 3 enzymes we talked about earlier. And it gets worse. When they hit these cells with inflammatory triggers, their ability to release the alarm cytokine IL-6 was severely blunted. So not only can the cell not digest the Alzheimer's cargo, its backup plan to spit it out and sound the inflammatory alarm is also totally crippled. Which brings us to the overarching conclusion of this entire paper, perfectly summarized right here. Quote, SORL1 deficient microglia are highly capable of engulfing selected cargo, but their intracellular sorting system fails to provide lysosomes with fully functional degradative and release capacity. They eat the amyloid, sure, but without that SORLA cargo manager, the delivery trucks break down, the enzymes get lost, the garbage piles up, and the immune response just goes completely dark.
This step-by-step molecular breakdown leaves us with a really profound thought to end on. If the missing CMPR delivery trucks are the primary bottleneck here, and if restoring CMPR could rescue this degradation pathway, could we one day develop therapies that fix these microscopic sorting systems in Alzheimer's patients before the irreversible damage is done? It's a question that completely pushes the boundaries of how we view neurodegeneration, moving from just treating symptoms to actually repairing the cell's fundamental logistics. Thank you so much for joining me on this deep dive.